Hi, everyone. Thank you for joining us for Abbott Labs webinar on their new point of care rapid test for COVID-19. We have a panel from Abbott that will explain the science and technology behind this new test and how it can fit into urgent care. The panel consists of Dee Chapman. Dee serves as the Strategic Business Executive for Abbott Rapid Diagnostics with oversight to the urgent care market. In her role, she manages the portfolio for both the cardiometabolic and infectious disease products. The development of this organizational role has offered Abbott the opportunity to learn more about the ways they can serve the urgent care market space, most recently with the funding of the diabetic study and now with the involvement in the antibiotic stewardship campaign. Her responsibilities include sales support, strategic account planning, and business development with the scope of urgent on-demand sites. She has served in the role during the she has served in several roles during the last 24 years within the Alere organization prior to the acquisition by Abbott. She has served on the Sales Advisory Council and won the President's Club over 10 times. We also have Justin Volk. Justin serves as a strategic business executive for Abbott Rapid Diagnostics with oversight in the urgent care market. In his role, he manages the portfolio for both cardiometabolic and infectious disease products. His responsibilities include sales support, strategic account planning, business development with all scope of urgent care on-demand sites. Justin has currently been with Abbott for 10 months and has been working in the infectious disease diagnostic market for over 8 years. And lastly, we have Dr. Norman Moore. Dr. Moore received his bachelor's degree in biology and physiology from Dartmouth College and his PhD in microbiology from the University of New Hampshire. Among other essays, he developed the first ever rapid test for Legionella and S. pneumonia, both which are now recommended by the Infectious Disease Society of America. I may have mispronounced that first one, so I apologize. After being an R&D director for many, many years, he took the role of director of scientific affairs, where he works with the medical community. He served on multiple NIAID grant committees, the CDC guideline group for rapid influenza testing, the College of American Pathology and Clinical Laboratory Standards point of care committees, and the CLSI's Infectious Disease Guideline Writing Committee. He currently has 6 patents and numerous publications and presentations. So before I go and hand it over to Dee, I just want to let you know if you have questions, you can go ahead and type those into the question box. We're going to go through the presentation, and then Dee will come back on to answer and take a look at the questions that you guys have. And then also this will be recorded, and you guys will receive a recording in your email tomorrow. So I will go ahead and Dee.
Good afternoon. This is Dee Chapman. Thank you so very much. We at Abbott are excited about the opportunity to talk with the Urgent Care Association about our ID Now molecular platform. We received emergency use authorization from the FDA on Friday afternoon for our COVID-19 assay, and it has been a whirlwind since then with the amount of interest around the country in gaining access to the test. We even got a little free press from the presidential press conferences, specifically on Monday when the president actually unboxed the instrument and displayed for all of us that how, as it was being detailed, how it was going to improve patient abilities to getting testing done at the point of care. Urgent care was even called out. We won't discuss that the instrument was upside down, but we were grateful for the free press. It is my pleasure to introduce Dr. Norm Moore. He's the Director of Infectious Disease. You heard his profile for Scientific Affairs, and he will walk you through all the clinical data. And then after that, we'll open up for questions that we can field for y'all. So thank you so much. Dr. Moore?
Okay, thank you. And I just kind of want to start off. We've got a little slight technical glitch. I can't see the slides, so I'm going to kind of go through mine, and hopefully we're going to be on the same slide with that. But I kind of probably want to start off with maybe a few questions that I've been getting, because basically we are in unprecedented times. And so quite often one of the first questions I get is, what are we going to be expecting out of this? And unfortunately, it's not a question that anybody knows. I will say that I'm on calls with the CDC every single Friday. They're giving me as much information as soon as they know it. If anybody has any questions, that is still the best website to go for the latest information. What we are now wondering is it going to follow something like previously to a former influenza pattern? So back in 2009, when we had that flu pandemic, we actually had it kind of roaring right through the summertime. And it really didn't quite take a break. We don't know necessarily if it's going to do that. It doesn't look like this virus does great in humidity, but obviously Australia's having a bit of an issue right now. If I look back at like the 1917 pandemic, which really was, the big one for influenza, that kind of took a break in the summer and then kind of came back in the fall. So I can't tell you what's going to happen, but I know that we're going to be dealing with it for a bit more time. We don't know what the model is going to be, when the peak is going to be. Again, there's a lot of models out there. I haven't really seen any models that have anything peaking in less than a month. But we are getting a little bit of information that some of the social distancing is working. I've been getting questions like, can you get it multiple times? That's still something we don't necessarily fully know. Certainly, a lot of groups are hopeful that you will build up immunity to it, and we don't know whether that immunity is good for 3 years or a lifetime or whatever it may be, but that is still a question out there. But the hope is that actually those people will be the safer ones once they've had it. I've been getting questions on ID Now whether we need to do testing under a hood. This is going to be a little bit unique, and this has been a bit groundbreaking for the FDA. Dee said that we talked about getting that emergency use authorization. What that means is that we are temporarily approved like every other test, but we were approved for near use to the patient, meaning that the FDA has wanted this test to be able to be used closer, because as we've all seen, a lot of these tests just aren't getting results back in time. And so we have that issue. Do we isolate the patient? If we're going to be sending them to the hospital, do we cohort? We clearly don't want them mixing with the elderly, because that has been our significantly biggest risk group, or any other risk group. So there are issues there. And of course, if we are able to test, I will say this, the symptoms have been all over the place. So at least giving that peace of mind, because we are now entering allergy season, and allergies, mild colds, more severe respiratory ailments, they're all blending in, and there are certainly patients that don't have any fever, just have a body ache. So it's become a unique challenge, and therefore to bring testing close to the patient is going to be able to help everybody out. So about that CLIA waiver, right now it is not CLIA waived, it is approved for near patients. What is the difference? The difference being that since this is not a fully FDA approved, like none of them are, it is like CLIA waiver, but the FDA is again doing something unprecedented by saying, yes, you are allowed to use it near that patient. Does it need to be done under a hood? The answer right now per the CDC is no. What they're saying is that if you have a potential coronavirus patient, you don't need that under the hood. However, you probably want to use the best protective equipment. I will say this, when we are talking about anything to do with testing patients, the actual most infectious part is actually that sample collection, and I could potentially talk about that later for people that need it. The test right now is approved for nasopharyngeal, nasal, and throat, and there is a lot of interest by the CDC on a potentially even a nasal, a self-collected nasal, because again, we want to start protecting a lot of the people collecting the sample. So kind of with that, I'm going to maybe start slowing down on questions and start getting into a bit more of the test itself. I'm actually borrowing the marketing person slides because my science slides haven't been approved because everything keeps changing so quickly, but I certainly can answer any questions. We don't expect a vaccine coming next year. Maybe after next season, but certainly not for next season, which means that not only we're going to be dealing with COVID-19 this summer, we're going to have to deal with the potential that people coming in this fall could have COVID-19 and influenza, so that's going to bring out its own wrinkles. So I hope everybody is okay with that. And with that, I'm going to assume we're on a slide that says ID Now Molecular Technology Review. Is that fair to say?
Yep.
Yes, we are.
Okay. So we're going to go to the slide that says the analysis of DNA or RNA for diagnostic purposes. With that, I'm going to say, basically, you can break down tests into two different groups, where we look for the virus itself, or we look for the body's response to the virus. And so right now, the biggest push is for looking for the virus itself. And when you look for the virus itself, the big tests out there right now are these, what's called molecular amplification. PCR is the one that people have been talking about for a long time, since that was the first one identified in the early 1970s by Kary Mullis. But they're basically all doing the same thing. We start with a specimen, we're able to break apart that virus, get that DNA or RNA out. In this case, it's RNA. We're going to make the RNA into DNA. We're going to be able to amplify that up. PCR does it by heating and cooling with these little primers. Isothermal uses an enzyme to do that instead. We can talk about that in a second. But the whole idea is it may be very hard to see hundreds of viruses, but it's very easy to see hundreds of millions of these little sequences. And that's what we do with this molecular amplification. It's literally get hundreds of millions of copies so that we're able to see that very readily. And that's what gives us the improved sensitivity and specificity. That will remind me, people are going to ask on any of the tests that are going to be available out there, what is the clinical sensitivity and specificity? And the answer is we don't have that right now. Under emergency use authorization, what we've done is work with the CDC and FDA to get an analytical claim. We know how many genomic equivalents that we see. So in the ID Now influenza test, we've been able to see 150 genome copies. So basically like 150 of that virus was there. We can see that and able to grow that up. What that means to clinical sensitivity and specificity is a claim, and we would have to wait for the FDA to issue that sort of thing as time goes on. So we'll hold off on that. But again, I've got the DNA, amplify it up, and then that instrument is going to be able to read that. So the whole issue is we are trying to get that as fast as possible. So next slide, number 4. Or the one coming after that, why molecular? Hopefully it's called the power of sample amplification. If I'm out there giving lectures, one of the bigger issues out there is sometimes I see that sometimes it's not the best clinical sample taken. And so if you've got the traditional lateral flow and you don't have a good sample, it's really not as able to overcome that as well as something that can be amplified up. So if I click that one more time, what I'm seeing is you can potentially get a false negative with a lateral flow if there's not enough there. And if I click it again, I'm actually amplifying that up. So hopefully we'll have a bit more of that amplification so we get those true positives. I will say that right now it is going to be helpful, and what we're going to be trying to be doing is making sure, whether it be through videos or little pieces, that people understand the proper way to collect, especially a nasal or a throat swab. You're not just rimming the outer nostril, it would all go up to the turbinate. So as long as people are doing that the right way, they'll be getting much better results than just something quick and easy. I can talk more about that later on if people want, but I'll assume that's okay. I'm going to go all the way down to where it says molecular technology umbrella. Just to kind of say, there's now a lot of new ways to do some of this ultra-sensitive testing. We've probably all heard of, again, what's called PCR, polymerase chain reaction. That's been out again since the 1970s. And the whole issue with that is if I take that sample and I heat it up, I can break apart the DNA, add my primers, and get that chemical reaction going so that it used to take these lab-based tests, would have exquisite performance, but it would take an hour or 2 hours, and it would have to be by a dedicated person in a dedicated room. Things have gotten easier. They have gotten, at this point, clear waived. And what we're now able to do is shorten that time dramatically. And in this case, rather than the traditional PCR where I'm heating and cooling, I am now doing things at one temperature. And because I'm able to do things at one temperature with the ID now, I'm able to shorten that time dramatically. So what we're seeing at this point is a negative result is in 13 minutes, and a positive result can be in as little as 5 minutes. So the whole hope here is we're trying to get what's happening with the patient mimic to what can be happening right there at that urgent care to make sure we're getting really the results while they're there to make sure we know and do the right thing for them. So because of that, things are now called all molecular diagnostics, and all of the new guidelines are reflecting that so that everything's under that same umbrella. So whether it's the ID Now test or traditional PCR, they're all considered the same. Molecular technology is defined. There's a bunch of different companies out there. I really won't worry too much about that. If anybody wants to talk about that, I can talk about that later on. But again, I'm just saying there's a lot of ways to get to the same result of amplifying that DNA up to get that sensitivity. So I'll leave it at that. Extraordinary technology. I'm going to then go to what is extraordinary about the ID Now. And yes, I'm a scientist reading marketing slides, so I hope you bear with me on that. What is extraordinary about the ID Now? Again, what we've tried to do is by getting rid of that heat, we're getting that speed to results down. We've tried to get it, again, under that 13 minutes. And some people get kind of like, well, how does that work? And I will quite often give the example, every living thing on the planet makes its own DNA at one temperature. We do it with enzymes, so other companies now are finding a way to also do it with enzymes so that we then can shorten all that time out, because these enzymes are quite quick once they actually work. If anybody, on the next slide, isothermal and PCR, if anybody's, again, confused, concerned about that, it's really most of the technology is the same. We still start with our target. In this case, it's an RNA. We are still using the same way to detect it. We have this little molecular beacon. It's a little piece of DNA that will actually light up if it binds. I know that sounds a little odd, but we have this ID Now machine that will actually sit there measuring. And if all of a sudden you get that exponential increase of this target coming up, you'll see that light get bigger and bigger and bigger, and it'll actually call it a positive, and we can see it that way. So I don't think we need to worry too much more about that, but just to say everything is basically traditionally the same between PCR and these isothermal techniques, with the exception of how do I open up that DNA to begin with. ID Now summary. Again, I know this looks a little bit commercial, but the whole idea is when we look for respiratory viruses for all these years, I made the BinaxNOW flu test, probably 20 years ago, and it had very rapid results. We were able to define who had the test, and if it was positive, you knew you were done. It had great specificity. If it was negative, sometimes you had to do additional testing to confirm it or look at the patient, but it had all of that, ability to, again, triage right then and there with the patient sitting there. The lab-based moleculars, again, offer that exquisite sensitivity, but it wasn't as actionable a result. The whole point with ID Now is trying to get that high-level accuracy with the ability to decentralize it and get that rapid result. So hopefully we're getting the best of all worlds. If I go to slide 15, I won't spend too much time on this because I'd really like to leave time for questions. The whole point of this is, luckily we had this technology out. So we had come out with influenza already, strep A. When that test is out there, there's no need to confirm positive or negatives on either or any of these. So either a positive or negative result is confirmatory and you're done. So those tests exist, and what we've been able to do is the whole point in how we've been able to get this ID Now COVID-19 test out in this short a time is we've taken the science, the technology we've learned from all that, and just pushed it in. So we have all the instruments available, we have all the manufacturing equipment available, and now we're basically switching it all to COVID-19 as we really try as best we can to help with this unique pandemic. On the ID Now procedure summary, this is the first test that our flu was the first one that actually was molecular and was CLIA waived. And so what you'll see with this, if people haven't seen how ID Now operates, it's a very, very easy to run GUI interface where it's just nice, pretty pictures. So if you don't even speak English, you can still run this test. Basically, you can scan that patient ID. So we've got a little scanner with that. You open the lid. We've got these, almost looks like little Tinker toys or what, Legos or whatever people want to call them. It's all color coded. So the orange goes in the orange spot, the blue goes in the blue spot. Very simple. It'll heat up. I take that patient sample. I'm going to put it in that little blue liquid. It twirls around there. That's going to take the virus. It's going to go in there, break it apart. I then have a little transfer cartridge that moves it over to the orange, and that's where the action happens. That's where the DNA gets replicated, and we're able to see it. So it goes into the orange, you close the lid, and you wait, and everything's automated at that point where it's going to show up as positive as, again, early as 5 minutes, or to get the full sensitivity out of it, the negatives will go all the way out to 13 minutes. On the next slide, the timely benefits of our technology. I will then go to time-sensitive care settings. Again, you guys are front lines, and I obviously can't thank you enough for all this, but we all understand that if we don't have a timely test, it's really not going to have the patient impact that we had before. And I will say right now, with things being unprecedented, I've seen a lot of unique ideas out there where, one of the local pediatric offices will actually say, okay, call when you get there. We'll send somebody out with a swab. They may not even necessarily have to get out of the car. They've even purchased tents so we can actually keep the lines separate. So if anybody's worried about COVID testing, they can be in one area. It's separated from patients in another area to, again, make sure that we're not really exposing the ones. And again, I keep mentioning the elderly, but they are usually the ones most at risk. When I look at Italy and how they've had the most massive problem yet of everyone, it's primarily because Italy is the oldest of all the European countries. And so that is the concern, making sure that we are as safe as possible with that. So again, we're under that 15 minutes. We can give the result right there. We can maybe even have them taking their own nasal swabs. We can set them up in a separate room. This is a very portable, I think it's like 6.5 pounds per instrument. So it's nice, easy, portable, and can be brought there. It can be done not only in urgent care, but in emergency departments. Again, we can separate those people out. And if they need to cohort, they'll cohort, send them home, whatever it may be that they need to do. On the next one, respiratory testing for infectious disease requires a different approach. I won't spend too much time on this, but just to say, again, the symptoms are unique in that they're so, I've never seen something quite like this where one person is aching, doesn't have a fever, another person has a fever, another person is coughing. It's really been trouble. I mean, I am called on to, and obviously I'm not a medical doctor, I'm a PhD. People could be asking, and we really can't decide anything without a diagnostic test because it is so varied. Again, here we are hitting allergy season, and you're going to get a lot of panicked people coming into your facilities that may just have an allergy and then can't differentiate that. So there's a lot of good reasons why, at least with something, getting that test out, we're going to have the best results. I will say this too. One of the best outcomes in countries has been South Korea. South Korea did so well because they really pushed a lot of testing and were able to really isolate the people that were positive much earlier. And so hopefully we can do the same as we are trying to live through this day today, where we want to make sure that if you've got a cold symptom, you're tested rather than going to, pick up more groceries. We've really got to make sure that testing happens and we really isolate those people. Respiratory testing sent to a lab. I don't have to convince this group of how you guys, being the front line, you need those results in a faster time. Right now, I am seeing many issues where if you're sending it off, it is taking hours and more likely days to get the result back, and we really don't know what to do with that patient. And especially right now, there hasn't been a good treatment. I won't comment on the malaria drugs that are potentially being approved for that. That I'm staying out of that one entirely. But at least with something like this, we know at least to tell the patient so that they know that they have it, or at least have that comfort not to. Time-sensitive care settings, emergency room, urgent care. Again, I won't spend too much time on that. If anybody needs any more information, again, the whole point is we're not doing anybody a favor if it takes too long. We have to have those patients waiting. We have to really try to make sure we triage them while they're there. Time results, timely decisions. If we're at that point, again, there's a lot of things that happen. And if I'm giving lectures out there, this is the kind of a great slide to talk about, because obviously speed to treatment will help with that. If we don't have, I don't know how many people are going to be taking these anti-malarial drugs. We could have shortages. There's obviously these drugs do have some side effects to it as well. So certainly we don't want people taking them that are negative. There's a lot of issues with that. We don't have to talk about antibiotic stewardship. There is no, at this point, specific antiviral medication. But I will say we may run into that point where come this fall, we're going to have to potentially be worrying about people that are co-infected, influenza and coronavirus. And at that point, we're still going to be looking to the CDC whether you test for influenza first because that is more treatable, or whether you want to test coronavirus first. A lot of that's going to depend on really the number of coronavirus cases still going on. So that's unfortunately a wait and see. Appropriate admissions, appropriate use of other testing. At this point, the assumption is going to be if they're coming in with a respiratory infection, since our influenza season has been waning down a bit, if they're positive for COVID, we at least know. And since our hospitals are being overwhelmed, that may be at this point, the doctor is going to make that decision to maybe at this point you just want to self-isolate rather than go into a hospital if you can at least take care of yourself. Again, those are all the medical decisions that I will stay out of, or I will get into trouble being a PhD rather than an MD. I don't have to talk about efficient testing resources. We are going to be stretched. We are already stretched. So the whole hope here is this is a far more useful tool so that we can make sure that we are getting that result as fast as possible. ID Now is the next slide. It is isothermal detection like I talked about. I think we've covered that. Again, the whole point is taking as much time away from that heating and cooling so we can getting that fast result. We get that identification. It is rapid. It is easy to use and minimally CLIA-waived in the other types of assays. Again, it is near patient for this one. ID Now, again, another marketing slide. Sorry about that. ID Now is molecular, meaning that we are looking for the, in this case, RNA and amplifying that up. So again, the whole point, getting hopefully that lab quality result now down to the patient. I hope that is okay. And with that, I was going to try to open it up more for questions than anything else. So I think Dee's going to be taking them. I can't see the questions, so I'm going to let that happen. And the questions I can answer, I can. Like I said, I'm on the call with the FDA, CDC as much as possible, but there's a lot we don't know. Hello?
Norm, can you hear me?
Oh, thank God. I actually one time gave a webinar, and 20 minutes in, somebody finally texted to say that it had shut off, and I've been talking to nobody for the entire 20 minutes. So thank God somebody's here.
Yes, we're here. So can you hear me now? You ready?
Yes. Yep.
All right. So I've seen a lot of questions on sensitivity and specificity. So if you go into a little deeper on the explanation of the emergency use authorization.
That is a great, great question, because that's going to come up a lot. What is the sensitivity to any of these tests? What we did, the CDC made available a thing called the genomic equivalence. Think of it almost as like how many viruses can you see, similar to that. So in this particular test, we were able to see 125 genomic equivalents, which is amounting to 125 per mil. So that's what the virus can see right now. What does that mean clinically? And clinically, we're not fully sure. I know that makes people anxious, but this is a virus that, when I'm really talking about influenza, quite often that peaks within the first 2 days. Coronavirus is going to peak maybe around day 5 or so. So we can't give a specific number. We can't say it's going to be 99% sensitive. We can't say 80. We don't know. And I completely agree with the FDA on this one that we can't say until we know. But again, at least with the molecular tests, that is a technology that has been approved by the IDSA on all of the newer guidelines. So I wish I could give a stronger answer. It may be a while before we know. So we're going to, again, keep with the FDA, keep with the CDC, and get the latest information as best we can.
All right, that sounds good. And this one I'll take. There seems to be quite a few questions about people wanting to know how they can get it, when they can get it, and where they can get it. So, and unfortunately, with the demand in the country at this point, we are in an allocation process to begin with. We are being given direction from the government and the highest level of Abbott leadership team on placing in the areas with the highest patient population and prevalence of disease state. So that is the first order of business right now, and that's obviously we've got 18,000 instruments that are in place around the country, and with the production capabilities at 50,000 tests a day, it doesn't take you long to figure out the math that we can't produce enough on the start. So we will be approaching on a very cautious the market that is going to get it, so that we can make sure that we can continue to supply their needs, and then we will be opening it up as the supply chain gets bigger. The other ones that I'm seeing on here, Norm, is symptoms may peak. Oh gosh, sorry, they're coming in really rapidly. Lots of questions.
There is a lot. This is tough. Yep.
Yep. There was a question on the. So is it a 13-minute test? So if you'll clarify there on the negative versus positive. I think people seem to have missed that.
Absolutely. Since the instrument is able to monitor the reaction as it goes, if it's positive, some of the positives have been coming up around that 5-minute mark, but to get the full sensitivity, it goes out to 13 minutes. So at that point, if you've got one instrument, you can do 4 an hour or more, depending on how many positives you have. If you have 2 instruments, obviously you can double that. So right now, the full negative would take 30. If it's positive, you're going to get 5, 6 on average.
Yep. Great. The other question, there is question about the sample that is allowed for it, and if there is a difference between the specificity between the three, actually.
That is a fantastic question. And again, what we've tried to do, the question is, all right, when the CDC first came out, they said, let's go nasal pharyngeal. And then they said, well, all right, maybe we'll do oral, the oral pharyngeal, meaning the throat swab, and now they're allowing nasal. There is no significant information right now that's going to be able to help define what the difference is between nasopharyngeal and nasal. I have gone all the way back to the Chinese records when this first came out. It looks right now that there's more receptors, so it may be that you have a better nasopharyngeal than throat, but that was one study in China, and that's not full. So here I am kind of giving a long answer to say we fully don't know. There is a publication that we are expecting this week from Washington State that may be the first of its kind that comes out, where the researcher said he's got good data on nasal swabs compared to nasopharyngeal, but he's not giving us a full recommendation either. So I am waiting patiently and even hoping that it is today, tomorrow. I didn't see it yesterday, so that, as far as I know, that's the first of its kind going back to this COVID outbreak. So hopefully more on that this week.
All right. Does a positive test need confirmation with a send-out?
It does not, per the FDA. So positive or negative, at this point, you're done. I would say make sure, I mean, sometimes people are coming in asymptomatically, and that certainly may be earlier than is going to be able to be allowed for this, for any of these tests. So just because they're asymptomatic one day, and if they get symptoms, that doesn't mean that they're still negative, if that makes sense.
Yeah.
If they're negative and they all of a sudden they get symptoms, they should be retested.
Perfect. All right. Good Lord. Lots of questions.
And I will say this while Dee is looking at questions. I'm trying to keep up with the CDC. If you have anything else, we try to be available. But the CDC is still the best website out there than anything else I've seen. They've really been excellent in updating.
All right, so we're getting a lot of questions on a comment that was made earlier in the presentation regarding the EUA and the language on moderate complexity and the addition site. So if you can clarify on that, on the FDA not issuing a CLIA status. There's
awesome.
A lot of
again, that is a very good, and I know that the College of American Pathology is actually meeting probably right now. Before I got on this call, I was texting with a person on the CAP call. So again, what we're trying to do is now make the government agencies work together, which is a bit unique. The FDA has said, yes, we want this near patient. Again, the whole point of what's called CLIA waiver is it has to be on an approved instrument, and this is an emergency use authorization. So this is the first time that the FDA has said, okay, we're going to allow a near patient without even an approved, a full FDA approval. So we're all in new ground here. So I hope that kind of people understand that what the FDA has said. I'm going to try to get more guidance from that, because here I am talking to these groups and making sure that they and CMS are in the same camp. And again, College of American Pathology is meeting today, so we should be getting more information on that out. As you can see, everything changes day to day. This is amazingly unique.
All right. And Norm, have you got the descriptor there of what it actually said for those that might not have seen what the approval was?
I can try to pull that up, but like I said, the FDA allows for near use.
Sure. There were some specific questions on what it said. I got it. It asked about supplies supplied in the kit. While we're looking for this, I can tell you that we will have included in the kit will be the throat swab, which is also used as the nasal swab. Those are included in the kit, and there won't be a need for additional swabs unless you want nasopharyngeal, and then those will have to be purchased outside of the kit. Someone asked about the patient fact sheet. Once they would tested positive, what are the directions as far as patients at that point?
That's really not up to a company. That's going to be up to, obviously follow what the CDC says, but if they're positive, that does mean that they have the virus and should be isolated, or whatever comes in with that. But I don't think we're going to be allowed to give any more guidance than positive, negative. It's up to the physician what to do. I wish I could say more, but that's I probably have my knees broken if I tried that.
All right. So the language is the ID Now COVID-19 assay is also authorized to be distributed and used in patient care settings outside of the clinical laboratory environment. That is the wording on it.
Yep.
All right. Let me make sure that we've answered all the questions. Will this test be a dual positive for influenza and COVID? Think you can answer that?
Right now, no.
That?
We are totally focused on COVID at this particular point. And again, you usually don't have to test for influenza during the summer times unless you're in like Hawaii, kind of get some more. But as we expect, right now it's COVID, and we'll see what the CDC recommends come next October, November. But right now, we're full guns on just COVID.
All right, and then here's one that's just come in. Is this tested IgM antibody test or an antigen test? And it goes on to elaborate on the antibodies, and I think you can answer that one pretty quickly.
Yes, it is not antibody or antigen. It is molecular. There are going to be some serological tests, meaning that rather than looking for the virus itself, I'm looking for the body's response. And so those, if you see a finger stick test coming out, that will be looking for IgG, IgM. The positive is that a finger stick is fairly easy to get. It's not going to be as sensitive in the earliest of infections as a molecular test, but they might have other uses later on. Are we going to be looking at those sort of serological tests? If somebody's positive next October, we may have some issues because is it a current infection, or did they get it in the summertime and it was so mild and now they have flu? But maybe we want to even look, and here I am speculating, I probably shouldn't. Maybe we want to start testing healthy people in the fall to see if they got it sometime between now and then, and they may be our safest people. But again, that's all speculation. Don't trust anything I say on that until we look at the CDC on how they want us to define serological testing later.
Good. The question is there a CPT code that is applicable for this test? Yes, the code is 87635. CMS has just written this code specific to molecular testing for COVID, and it's supposed to reflect today on the fee schedule. Norm, you got anything to add on that one?
Nope.
Okay. From your experience, would you have confidence in the results of this test?
We have confidence, and we have tons of data on the three assays we have. We have, again, taken all that technology, so the technology is going to be very, very similar to our flu test and the latest version of flu. That's all I can say right now is we've got confidence in the technology itself. I can't guarantee 100%. Nobody can. Again, this is unprecedented.
All right, next question. Does the test differentiate between COVID-19 and other coronavirus strains?
We do not want to detect other coronavirus strains. This is only for COVID-19. The issue being a lot of the other coronaviruses may cause just a common cold. And so, yes, you'll have some exceptions to that. But for this, we only want to be detecting COVID-19. And any test that you buy right now, it is better to detect only COVID-19. And I hope that makes sense. I can go into that more, but right now, yes, we don't want to detect any other ones.
Okay. How long must the patient have been symptomatic before they can test positive?
Great question, and we're going to see. I mean, again, it's got a different. Again, I wish I could say, what does the CDC know? We've been fighting back and forth. Even how long is a person, infected before they actually show symptoms? We are fighting a lot of battles right now in trying to get this information. So there is no data to say on day one you're going to have this sensitivity, day two you're going to have that sensitivity. It's all brand new.
Okay. Here's one I think that what you'll find interesting. How does this compare with current COVID-19 point-of-care testing being marketed, which is checking for IgG and IgM titers? Would combining these tests with the ID Now test increase the sensitivity and specificity?
A good question. The assumption right now is that an IgG is usually going to have great sensitivity later in infection. The assumption right now by everybody is that the molecular tests are going to have much better sensitivity early on, because it takes quite often several days for you to develop an IgM and then after that an IgG. So right now it looks like this is a one-stop shop, but again, I think there are some potential interesting applications for the serology later on. Again, it could be interesting to see if people have been exposed. So there could be other applications later on, but I haven't seen that you've needed, and right now the FDA is saying something like a molecular test can be standalone.
Got it. I'm good. The questions continue to come in, and I'm assuming that we'll be able to answer some of these back to you in chat function. And there's quite a few that, again, we just don't have the capability to answer with EUA approval. But there's one here on the collection by a patient. Can you elaborate on what we're saying there?
Right now, the CDC, I think there's either a link in our package insert or we're providing a link, but the CDC is, again, I've never seen this before. The CDC is suggesting that, a good sample collection by the patient may be helpful, and especially when we're talking about an urgent care that maybe people are concerned about. I will say this, sample collection is one of the more difficult times, making sure that the provider stays safe. That patient can certainly cough. There's a lot, obviously there's a lot of things that can happen. So go to the CDC, and the CDC does have some information on a self-collected nasal. So I'm going to be very curious on that. I will say this, I would rather, if there is a self-collected nasal swab, that at least somebody sees a picture or sees a video or something, so they just don't rim the outer nostril. I want them to go up to the turbinate, and as long as they have that, that could be very interesting to see.
Okay, I think that, again, you know what, there are a couple of others that I think would be good. They may be premature because this is something that as the kits are launched within an account, they'll get this data. But this is a reportable information. This is how numbers are being reported into the CDC. So it was asking, do they have reportability responsibilities if they have ID Now tests for COVID that they're running?
I'm afraid to go to your state to make sure. I don't know if that's a state-to-state thing, but I don't want to trip anybody up on that and say the wrong thing.
All right. Again, they continue to come. I think we'll make the chat the answer back, because some of them are just repeat questions. So, Norm, if you got anything else additional, or
I will say this. I know I'm waffling a bit. When I'm on stage, usually talking about things like flu or other diseases, it is so much easier to have all the data behind me. We are all in a unique situation, so I hope that was okay putting up with this. Again, there's a lot we don't know, so things are changing. So good luck to you guys. I try to make myself available. I mean, obviously, I can't answer every question for everybody, but we're trying as best we can. So, reach out to your appropriate Abbott person. We can try to get information out to you. Good luck to everybody.
All right, I would just like to thank both Dee and Dr. Norman for, spending some time giving us this information. We also, as you guys know, if you don't know, there's the UCA has a coronavirus resource page. We will be posting this webinar on that page once we get the recording of it. So you would be able to share that. And then as far as communication back on any unanswered questions, Dee and I can work on that to get you those answers. So Thank you again for participating and all that you guys are doing out there, on the front line. So I will go ahead and complete this webinar for today. Thank you.
Take care.